Thank you. Hello everyone. My name is Huiying Yan, and I'm from the university. Today, let me introduce our work, GORetriever, ranking protein description-based GeoCandidates by literature-driven deep information retrieval for protein function notation. Yeah, that's a long title. First, as you can see, AFP, that is automated protein function prediction, is a large-scale and multi-label classification program that is in UniProt. There are more than 250 million proteins, and there are over 50,000 geolabels to describe the functions of proteins. However, only less than 0.1% of them have experimental functional annotations. So it's important to develop high-performance AFP methods. And on the other hand, to systematically describe functions, scientists develop GO, that is gene ontology, is tree structure and has three domains, MF, BP, and CC. It makes AFP more difficult. And the previous AFP methods, they regarded it as a multi-label classification task and use different data sources such as sequence, structure, network to predict the geoterms. And another way to solve AFP program is to incorporate different data sources. For example, NetGeo3, it uses, four sequence-based methods, one network-based method, and one test-based methods, and incorporated them with a learning-to-rank framework, and it achieved the state-of-the-art performance. However, this previous work, they didn't use the textual information well, but as you can see that in Swiss prot, about 82% of the proteins have literature information. However, just about 16% of them have function annotations, and a lot of them are annotated by human curators based on the relevant documents. On the other hand, the previous methods, they use textual information in a simple way. For example, the deep test to geo, they use the document to vector and TF-IDF to generate the test information, generate the test representation of the literature. And to mitigate the gap and to save the time and cost of human curators, we developed the GeoRetriever to annotate these proteins well. And the GeoRetriever, this is a two-stage information retrieval framework. As you can see that to solve the three challenges using literature data for better AFP, we developed three components. First, for the challenge one, how to construct an effective protein representation. We use the sentence extraction to obtain the informative sentences from annotated literature. And for the challenge two, how to conduct semantic matching between protein and geoterms, we developed Rerank to find the deep matching between candidate geo and the informative sentences. And for challenge three, how to make the whole procedure feasible, we use a retrieval component to candidate the geo from the similar proteins based on their description. And then let me introduce the detail of each component. First, sentence extraction. We use a sequence-to-sequence model. And so the input would be natural language, that is a query sentence, and to generate the relevance. And so the query could be, what is the branch description of protein? And the branch description can be replaced by MF, BP, or CC. So, and the sentence could be each sentence in the literature. So the output is the log likelihood probability of generating true in the next token. And after that, we reordering the sentence from each literature to form the informative sentences, and we obtain the top 50%. And note that the number of each letter in the of each the number of sentences from each letter is not fixed. That is to say, we may extract 3 sentences from one letter and 0 sentence from another. And then is the retrieval. We try to retrieve candidate geoterms from proteins in the training data with a similar description. As you can see that giving a target protein, we retrieve the similar proteins based on their textual information, that is the name and the species of each protein, and then we add the corresponding geoterms of each protein to form our final geocandidate set. And then the rank, after obtaining the informative sentences and the candidate geo, we calculate the relationship between them use a cross-encoder structure. And you can see that the input is the protein and the description, and it's also a natural language, and the another input is the geo definitions of each candidate geo, and then we obtain the token of COS, the representation of COS token as the final score, and then we order them to get the final geo sets. As for the data, we randomly selected 1,000 Swiss prot proteins for testing based on the species distribution of kappa-5 test superset. And the geo notations are from 3 databases, Swiss prot, GeoA, and Geo, with experimental evidence or just annotated by human curators. The textual information are from Swiss prot. We selected the protein name, change name, and organism name as the protein descriptions, and the title and the abstract of each PubMed identifiers as the literature information. Our sentence information are from UniProt. Now let's move on the main results. As you can see that, well, let me illustrate that the test proteins are these proteins in our testing set with literature information. And as you can see that GORetriever, it achieves the best performance in all 3 domains, especially for BPO. And the sequence-based methods are effective for MFO, which is difficult to describe just in literature. However, text information plays a crucial role in prediction functions on CCO. As you can see that both LR test and the GORetriever, they show a great performance. And then we focus on difficult proteins. That is proteins with blast identifier of less than 0.6 to any proteins in the training data. As you can see that most sequence-based methods show heavy performance decreases, especially BLAST KNN. However, our method GORetriever, it shows a slight decrease just from 0.619 to 0.614. And to better understand the role of each stage in our method, we develop our evaluation study. We replaced the 2-stage rank component. We replaced the first stage component retrieval with other methods such as LR-ISM and BLAST KNN. And in the first 2 lines, you can see that GORetriever is improved progressively during 2 stages. And in the last 4 lines, you can see that GORetriever enhances the performance on sequence-based methods via rank. And then to examine the performance of GORetriever, we tested on the kappa-5 supertest. And first, as you can see that compared to LR test, GORetriever shows a better performance. That is to say that GORetriever can make good use of textual information on proteins and geoterms in a reasonable and efficient way. And however, the poor performance behind this is that, just about 90% of proteins in kappa-5 test set, it has literature information, and our method, it cannot predict them well. However, after incorporating GORetriever with NetGeo3. It shows a great performance. You can see that it just behind the GeoCurator the first place, and it can demonstrate that GeoRetriever can complement the traditional AFP methods. And then we select one protein to understand the role of textual information in the case study. And as you can see that compared to the comparative methods, both the text-based methods, LR test, GeoRetriever, and other methods with Rerank, they can correctly predict the developmental process branches well. And this result can clearly demonstrate the advantage of incorporating textual information for AFP. And then we select two geo terms to understand the textual information further. And as you can see that the bold, it shows the textual information, textual relationship between geo definition and relevant sentences, and you see that we can capture the relationship well. So in summary, textual information is not well used in previous work. However, our method GeoRetriever can annotate the protein function accurately by using textual information, especially powerful for predicting difficult proteins. And besides, our two-stage retrieval-based framework, GeoRetriever, is effective for capturing textual relationship between proteins and the geo labels. And this work is supported by NSFC, and thanks to our advisor, Professor Zhu, and our collaborator, Professor Mamisuka. And thanks to our GeoCurator members, Shaojun Wang, Tianyang Huang, Hanchen Liu, Weiqi Zhan, and Wei Liu. And the following is the rated works of our lab to develop better AFP methods. If you are interested in it, just come and visit us at the Fudan University or contact Professor Zhu at this email. And thank you for listening, and any question?
Thank you. So we'll go first to an online question. Did you predict proteins using a single Go term? Do you have any constraints in this regard, such as requiring the protein to have at least 50 Go terms?
Did you predict proteins using a single Go? Well, sorry, we don't concern on it. We don't care about it. We just concern about the proteins. Not the geo term. Yes, we do not make constraint on the geo terms, the number of geo terms.
In the UniPro or UniPro GOA database, some PubMed citation are cited based just due to their methodology. For example, the most highly cited paper is called Phylogenetic-Based Propagation of Function Annotation within the Gene Ontology Consortium. And it is the paper cited for all the IBA evidence Go term annotation. So do you remove those kind of literature?
Yes. Your question is that these papers that are annotated by many proteins, do we remove them? Yeah.
Yes.
Remove it?
Yes.
Yes. We have did some further experiment on it, and we found that after removing it, the literature from Geo and Geo, it had be a lot, just a little, and the results is not shows a great decrease. So maybe the literature from Geo A and Geo is not important for our further predict, and as you can see that we developed the sentence extraction to extract the most informative sentences. So I can see that we are robust to this general paper. And that is also a good question. We are trying to develop an end-to-end system to annotate the literature and then predict the function. So maybe the program will be well solved in the future. And thank you for your advice.